So what's gene editing? Why people are so much interested in that? Long time back, like in around 2006, I visited a banana farmer in Uganda. I saw a big field, then entered into the small house of the farmer, and I saw like an old farmer sitting on the floor and very sad. So I talked to him, and then he said like, yes, we don't have food because his field is completely wiped off. And I said, how can I help you? So gene editing is a new breeding tool which has a lot of potential to accelerate the breeding for improving the varieties. And it is very precise method, so scientists can actually manipulate the genome of the plant they are interested in with little bit tweaking. Using the gene editing technologies, the scientists can actually either add, delete, or substitute. So gene edited products, if there is no foreign gene presence in them, they are actually treated as conventional, non-GM. So 4 countries in Africa, Nigeria, Kenya, Malawi, and Ghana, have now the guidelines approved according to which the gene edited products, when there is no foreign gene integration in them, are actually not treated as GM. I will say gene editing is not a new technology. It has been since ages, because nature has been always editing, and then the farmers were selecting. Later on, the scientist has actually developed the technology like talents, like CRISPR, which are more precise. And this area is really evolving very fast, because every time you see, even the CRISPR-Cas9 technology was first reported in plants in 2013, but now we have even more newer technology like base editing, prime editing. And because of the broad application of these technologies in the plants, in the animals, but also in the human for the gene therapy, this technology actually got the Nobel Prize in 2020. So at IITA, we started using this technology in 2015, and we started working on banana.
So banana cell suspension is highly desirable plant for the banana transformation, and we have about 10 cultivar banana cell suspension in our lab, and routinely we are developing new one cell suspension with several cultivar, and regularly we are using these cells to do banana transformation and genome editing.
So on our banana projects, actually there are some wild type of bananas which has natural resistance to the disease, but these bananas are not cultivated. So we have done a lot of molecular study on those wild type of diploid bananas, and then we are only transferring the information based on those wild type bananas to the farmer preferred cultivated varieties, and then we are trying to develop the disease resistance. Currently, we are working on the projects where we are targeting some of the genes we call the susceptibility genes, because these are the genes which are involved in developing the diseases. So we are making a specific, only few base pair changes in those genes, so that these genes are are not functional and they cannot develop the diseases in the end. And we call it, we are knocking out those genes. Quite a lot is already happening in Africa as well. Like our lab is working on bananas. We are also working on yam for mainly for the diseases and pest resistance. The key thing here is that before starting the project, we need to get the approval from the National Biosafety Authority for the use of the recombinant DNA in the contained laboratory and the greenhouse. So this is the Biosafety Level 2 facility, and here is only the people who have the access, means their projects are approved for doing the contained use facilities. They are the only people who are allowed to enter into this facility. So before starting the work, our projects are approved. As you can see here, this is for the gene editing of banana for the disease resistance. and this is another project for the viral diseases resistance. And then also here we have the control access, and only the people who are working on these projects have the control access, and we can actually open the door using this access and come in. So this is a double door facility for the containment. So when one door is closed, then only we are allowed to open the second door. So basically you need two things in order to do the genome editing. The first is the genome sequence of the crop variety you are interested in. And then you need the tissue culture transformation facility. So with the bioinformatics approach, that's where you identify the targets you want to edit. And then once you identify the target, you have to design the guide RNA, because guide RNA are the one which actually tells the endonuclease, which now in our case is the CRISPR-Cas9, where to make the cut. And once the double-stranded cut is done, actually the cell's own machinery uses it to repair, and during the repair, some mutations can happen, but these mutations are very targeted. So delivering into the plant cell, there are also various ways you can deliver. You can deliver either making it as a plasmid, which is a very traditional way of delivering into the plant cell, or you can also deliver Cas9 as protein and guide RNA as the RNA and make a complex, a ribonucleoprotein complex, and deliver into the plant cell. But whichever way, either of those two you use for the delivering, you still need the tissue culture transformation facility. This is our tissue culture lab, and we have intentionally separated the tissue culture lab from the molecular and the gene editing lab, because from the tissue culture lab we also use for the seed multiplication, and some of the times the seed can also go to the other partners, and so we don't want any contamination of any transgenic or the gene editing products into the tissue culture. So that's why we have separated, and even the people who are working into the tissue culture lab and the transformation lab, actually they have to wear different lab coats which are color coded. So the people, when they are in the tissue culture lab, they wear the green collared lab coats, and then the people who works into the molecular facility and for the gene editing, they have the blue colored lab coats. And we have very robust banana transformation system in our place based on the embryogenic cell suspension, because the beauty about the embryogenic cell suspension is that 1 ml subtle cell volume of embryogenic cell suspension can give us actually thousands of plants.
This is the banana cell suspension which I have to use for cell transformation experiment. You can see they are the very fine cell, and each cell has capacity to develop one genome edited plant. And this is the agrobacterium. This is called genetic engineer. And when this genetic engineer, we have to grow the liquid culture. After growing the liquid culture, we can make a pellet in the centrifuge. Then we mix the embryogenic cell suspension with the agrobacterium. And in this way we co-cultivate both together agrobacterium as well as cell suspension, and these agrobacterium go and insert the plasmid into the banana cell, and when we regenerate the plant in selection media, finally we get that genome edited plant. This is the first stage of the co-cultivation, you can see here, and they are very fine cells. Millions of cells are here. Each cell has capacity to develop the one genome edited plant. And after this stage, we have transfer the cell one by one in the another plate of regeneration. And here, because one plate have aggregate, so we have to isolate one by one. Here you can see the shoots are coming out, in the regeneration media. They have the hygromycin selection.
And these regenerated plants, now we have to actually characterize them to see the targeted mutation. And in order to detect the targeted mutation, we do the sequencing.
So what you have, or what you see here in the screen, is the data that we got from the lab, from the sequencing reactions. And these are the A's and T's and G's.
Once we do the Sanger sequencing, we align the sequences of the edited event with the non-edited event to check what sort of mutations we have got.
If there are any mutations, then you're able to confirm by comparing to the original sequence. So this is called alignment. So we align the original sequence with what we've been able to sequence, and you're able to see differences within your edited region or your target region of the gene. You can do that for multiple lines. For example, here you can see differences. So these are regions that are edited.
After getting green shoot in the plate, we go on the transfer them the proliferation media in the tube. And after tube, they have to go in the baby food jar. They are well rooted plant. From this stage, we take plant to the greenhouse for phenotyping.
So the trait we are working on is the disease resistance, so like a bacterial resistance. So we have to now challenge these plantlets with the bacterial infection, so that to see what is the response.
We transfer them to the glass house for acclimatization, but before then there is a form that we fill. We call it an internal transfer form, which will help us trace the plants from the lab to the greenhouse. So we fill that document at the internal transfer form, indicating the number of events, the construct, and the number of events that have been taken to the greenhouse for acclimatization. Then in the acclimatization process, we begin with small cups, and after a month, the small cups are placed in 1 month in the humid chamber in the greenhouse. After that 1 month, they are placed in bigger pots for 2 months, after which they are ready for disease screening in the glass house.
So in the successful promising edited event, which has already confirmed the mutation, those plants we find like they are resistant to the disease and we don't see any disease symptom in them, in comparison to the control non-edited plants, which will actually start showing the symptom after bacterial infection, like after 10-15 days after the infection, and then within 30 to 60 days the plant will completely die.
You can see the control plants, it's showing the symptom completely going to wilt in 60 days. And this plant, inoculated plant, you can see only inoculated leaf had died, and remaining plant is completely healthy and growing well. There is no disease symptom.
So we have developed Banana Tracker, which is a web-based app, and so it's starting from the creation of the cell suspension up to the end of the product. Each and every step is recorded. If we scan this barcode, then in that case we will know the whole history of this, what variety of banana it is, what genes has been used, where we have manipulation and who made this one and what date we have started the experiments. Everything can be captured using this barcode. IITA is a certified member of excellence through stewardship, and this facility is certified and it's annually audited by our internal auditors, and then every 3 years the facility operations are audited by certified members of the ETS. So all the people who are working in this lab, they are trained for the biosafety compliance and the stewardship, and we follow the standard operating procedures and manuals which are all placed in the lab. So we are using this platform to also train the scientists, and we have started in collaboration with UC Davis under the African Plant Breeders Academy CRISPR course. We have other partners like ICRAF, ILRI, and then it is funded by multi-donor, and under this program actually we want to train 50 to 80 African scientists within the period of 5 years, and we feel like this gene editing is a very promising a technology which has lot of potential, because right now in front of us the major challenge is how to feed the growing population. So we need food and nutrition security in our countries. One tool will not be able to solve all the problems. So gene editing has added one more tool in our toolbox which scientists can apply as per the need, complementing the other tools.